A student investigated the effect of enzyme concentration, that's how many enzymes there are in a solution, on the mass of product formed in 1 hour. So, if you remember, enzymes are proteins which have a very specific shape, or three-dimensional shape. They have a very important region called the active site, and that has a complementary shape to a specific target molecule we call a substrate. So to put it into real terms, for example, in your saliva, you have an enzyme called amylase, and it will act only on the substrate starch because they have a complementary shape, but amylase does not have a complementary shape to any other target molecule. Because of this specificity, because of the specific nature of enzymes, we call this model the lock and key model. So an enzyme will bond, or the active site of an enzyme will bond with a specific substrate. It will form an enzyme substrate complex, and then the enzyme will go to work helping the molecule break up very quickly to form products. So we're investigating the effect of enzyme concentration, how many enzymes there are, on the mass of product, the quantity of product formed in 1 hour. And you can see here the independent variable. What we're changing is the concentration of enzyme. Arbitrary units means you just don't have to worry, you just assume they're in some suitable unit. And you can see it's going up in steps of 5. So 5 concentration to 10 to 15 to 20. And then the dependent variable, what we're measuring, not the independent, what we're changing. Independent always goes in the first column, dependent in the next. Dependent, what we're measuring is the mass of product in grams, so 15, 22, 25. You can see this doesn't go up in the same sort of regular steps, but there is a pattern. It's going up, 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 and then it's leveling off, it's staying the same. So the first question says, complete the graph by plotting the points and drawing a line to show the trend, the pattern in the data. The first 3 points have been plotted for you, so you can see 5, 10 and 15 are there. Now the common mistake here made is firstly students do the plots but they forget to draw the line, so they miss out on that second mark. The second mistake is students think that a line has to be straight. They don't think a line can be curvy, but of course a line can be curvy. It just has to reflect the pattern in the graph. So you go from this point onwards, 20 to 40, and you plot 20 to 32. Remember, each one of these is 1, so it's a very easy graph to plot, 32. Then 38, 25, there's 38 there, and then 40, 40, 40. Just make sure you draw crosses rather than circles, because crosses allow you to be more accurate, and also it reflects what's happening already. Once you've done that, you draw a line of best fit. So this line has to go through a lot of the points, or the majority of the points, but doesn't necessarily have to go through all of them. So for example, you can see I've missed out that one. So it's just important that you reflect the pattern, and then you get 2 marks. Next is an easy question if you understand what it's asking you to do. It says describe the effect that the enzyme concentration has on the mass of product formed. In other words, describe the pattern that you see when you're looking at how the independent variable affects the dependent variable. A very common question. So if we look, all we have to do is describe the general pattern. So at first it increases, so the mass of product increases with increasing concentration, but after a while it levels off, and that would give you 2 marks. There's obviously 2 points or 2 things to say here. So let's have a look at the answer. Mass of product formed increases with enzyme concentration, 1 mark, and then levels off, 2 marks. Remember, describe means say what you see. It doesn't mean give a scientific reason for it. It doesn't say explain the effect that enzyme concentration has. Then you'd have to talk about enzymes and active sites and enzyme substrate complexes. But this is simple, just describe. The ratio of enzyme concentration to the mass of product formed using an enzyme concentration of 40 arbitrary units, or 40 units, is 1 to 1. So what does that mean? Well, it means that if you have 40 concentration or units of concentration, you also get 40 grams of product, 1 to 1. And the reason why you say it's 1 to 1 is they can both be simplified. If you divide them by the same amount, you can reduce them to 1. So for example, they're both divisible by 40, and by doing that, you'll get 1 for each, 1 to 1. But the question says calculate the ratio of enzyme concentration to product formed when the enzyme concentration is 5 arbitrary units. So how do you do that? So firstly, you look at 5 arbitrary units and you see that 15 grams is produced. So doing this is actually quite easy. What you need to do is you write, at the moment the ratio is 5 to 15. To work out ratios or simplify them, you basically have to divide both figures by the lowest of the two. So that would be 5. So you divide this one by 5 and this one by 5. So 5 divided by 5 is 1, because that brings everything down in terms of 1. And then 15 divided by 5 is 3. So that's the ratio, 1 to 3. The investigation used the enzyme pepsin from the stomach at a temperature of 37 degrees Celsius and at a pH of 7. So 37 degrees Celsius is basically optimum temperature for enzyme activity in the human body. What I mean by that, it's the temperature at which enzymes work fastest. pH 7 is usually the optimum pH, neutral pH, optimum pH in the body, because most places in the body are neutral, so it makes sense that enzymes work fastest in neutral conditions. However, in certain regions like the stomach, where the pH is much lower, then it makes sense that enzymes in the stomach have a different optimum pH. Their pH, or the most favourable pH for their activity, for them to work, is pH 2, which is the pH of hydrochloric acid in the stomach. So, the question says, which statement gives one way to increase the mass of product formed in this investigation? So let's go down one by one. You've got to think carefully. Firstly, increasing the pH. So increasing the pH, sometimes people immediately in their head, they think that means making it more acid, but it doesn't. What you're saying is it's pH 7. Increasing it means making it greater than 7, so 7 or 8 or 9. But of course, these aren't acid, these are alkali. And an enzyme that operates in the stomach is going to favour acidic conditions. So this would not work. Decrease the temperature. Well, this is optimum temperature where enzymes are working fastest. If you decrease the temperature, they'll have less energy and they'll work slower. So that doesn't make sense either. You've got to be careful. The other way may not work either, because optimum means best. If enzymes become very hot, they can denature and stop working as well. But I'll talk about that in a second. Next, decrease the enzyme concentration. So if you're trying to basically bring about an enzyme-controlled chemical reaction, if you take away the thing that's causing the reaction, you're not going to make that reaction happen faster. So decreasing the enzyme concentration doesn't work. So we're left with 1. We know it's the right answer already by process of elimination. But increasing the substrate concentration, well, that makes sense. If we're basically putting more of this stuff in there, then we're increasing the chances of an enzyme colliding with it to break it down or to help break it down to make product. So we're increasing the frequency of successful collisions between the enzyme and the substrate. So that is the answer. So another question which requires scientific knowledge now. Explain means you're going to have to give a reason. Explain why a temperature of 80 degrees Celsius was not used in this investigation. So I've already talked about how enzymes have a specific active site and they work at an optimum temperature, right? If you go above this temperature, the bonds that hold the enzyme together start to break, and that can change the shape of the active site and the enzyme. That means that the active site is no longer complementary to the substrate upon which the enzyme acts. So this stops the enzyme working. This act of changing an enzyme's shape is called denaturing, and it can happen through increasing the temperature or changing the pH. So, but not lowering the temperature, only increasing the temperature, because you're giving the bonds more energy to break. All that happens when you lower temperatures, the enzymes work slower. So explain why a temperature of 80 degrees was not used in this investigation. Well, for enzymes to work, they must form enzyme substrate complexes, and that works by them having a specific shape. So when you increase the temperature to 80 degrees, it will denature the enzymes. That means it will change shape. Now, why does that mean they don't work? Which changes the shape of the active site, so no enzyme substrate complexes form. Quick correction. So no enzyme substrate complexes form.